This is a presentation on the importance of evolutionary processes in heliconius butterflies on speciation, and my name is Brooke Sorenson. So in my literature research, I found that hybridization in heliconius butterflies supports the claim that hybridization in animals leads to speciation, and therefore has an evolutionary role. And so the definition of species is something brought up in the evolutionary world quite a bit, but it's also, and it's one of the most important things, but it's also very complicated and highly debated. And so I dove a little bit into looking at that. And a key to understanding the definition of species is determining the evolutionary processes that drive divergence. And that divergence leads to speciation. And so a major source, and so scientists have found that natural selection is the primary driving force of evolution and driving divergence, but it's important to look first at the variation in the gene pool that this variation allows for things to select for or select against, allows natural selection to happen. And so it's just as important to look at as natural selection. And so where does this variation come from? Well, a major source of this variation is hybridization. And so what hybridization is it's an interbreeding between distinct populations. So an interbreeding between species or varieties. And so the type of hybridization I looked at in my study on the butterflies was, in my research on the butterflies, was two allopatric species. And so allopatric are species that are geographically isolated, but then there's a restoration of symmetry. And so sympatric species are species that are not geographically isolated. And so when they're no longer geographically isolated, they're allowed to reproduce, and you can get this hybrid reproduction and formation of hybrid individuals seen here on the figure. And that can lead to speciation, which we will talk a lot about later on. And so hybridization is most easily recognized when these two allopatric populations come together, and there's that restoration of symmetry, which creates hybrid zones. And so you can see that in the image here. You see an initial population that was geographically isolated, and the isolated population diverged. They became separate species. Then there was a restoration of symmetry. They're allowed to reproduce, and it created hybrids in this hybrid zone. And these hybrid zones produce diverse genotypes that allow researchers to map genes that lead to speciation, map the genes that are leading to reproductive isolation. And these hybrid zones produce data that can't be made in a lab by crosses, because lab crosses only have a few generations of recombination, and so they don't have these diverse genotypes to study. And that's why hybridization is such interesting research to look at, and why I looked at it a little closer for this presentation. And so species boundary is important when looking at the definition of species, and it's a term that's frequently used in evolutionary biology. However, what exactly the species boundary is not always clear, and the definitions vary. Despite that, between the definitions, you can see one thing in common in the definitions, and that's that a species boundary is some sort of boundary that prevents gene flow between two populations. So you can see that in this first image here. You can see the genetic identity of two populations diverging, becoming increasingly separate from one another. And as that occurs, you eventually get to a point demonstrated here by this yellow X where they're subspecies of the same species, but they're still the same species. And then as you continue, you get to a point where they're entirely different species. In the time, in the points between those 2 points, is called the species boundary. And so oftentimes these boundaries are visualized in phylogenetic trees, which works well for species with old divergent times. And in that case, the boundary is defined by the genes that separate monophyletic groups. And so a monophyletic group is just a group of all the descendants of a common ancestor. And so you can see that highlighted here in yellow on this phylogenetic tree. However, this visualization of the species boundary using these phylogenetic trees doesn't work well for species that have recently diverged or are continuing to exchange genes, for example, in species that hybridize. And so hybridization is important in research and discussing the species definition and speciation. And so I looked at whether this hybridization contributes to speciation and actually has an important evolutionary role. And so hybrid speciation is when there's a formation of a new hybrid species, and that species is reproductively isolated from the parents. And so some people propose that hybridization actually makes two populations more similar, and therefore opposes divergence of the two populations and opposes speciation, and therefore doesn't really have a super important evolutionary role. But this hybrid speciation has recently become a possible mechanism for this in animals. And so I looked at two questions. Number one, does variation that leads to speciation come only from new mutations, or does hybridization actually play a role? And two, is speciation a clear boundary, or is it more like a continuum? And so looking at the first question, it's important to look at introgressive hybridization, which is an incorporation of alleles from one species into the gene pool of another species. So you can see that here in this image, you see species A and species B, and they hybridize and create an F1 generation of hybrids. And then this occurs across the species boundary. And then that F1 hybrids back cross and reproduce with one of the parent species. So in this case, they're back crossing with species B. And eventually the alleles from species A that were in those hybrids become incorporated into the gene pool of the other species, species B. And so this introgression may be important in speciation. So we can look at adaptive introgression, which is an introduction of these new alleles into the gene pool of another species leads to an increase in fitness in that species. So you can see that here in this image, the two species A and species B were geographically isolated and they diverged here. Then over time, there's a restoration of symmetry and they were able to reproduce and create hybrids. And integration occurred and there was an incorporation of species A alleles into the genome of species B. And these alleles led to an increase of fitness. And so species B diverged into a new species, species B prime, and this is speciation. And this is where the study of heliconius butterflies comes in. And so heliconius butterflies frequently hybridize across species boundaries, and genes encoding some of their wing patterns have done this introgression. They have introgressed. And this is seen in specifically the two species Heliconius melpomene and Heliconius sydno. And so Heliconius melpomene has almost 30 different color patterns depending on its geographic location, but they generally fall into two phenotypes: red bands seen here and rays seen here. And then Heliconius sydno is characterized by the phenotype of a yellow band. However, some subspecies under Heliconius sydno, Heliconius hirripa and Heliconius temerata, show phenotypes of red bands and red rays, which are seen in the Heliconius melpomene. And so it's hypothesized that those red bands come from adaptive integration with Heliconius melpomene. And this was debated at first, but later DNA sequencing evidence in Heliconius hirripa, seen here, supported that those red bands did come from adaptive integration between the two species. And so this adaptive integration between these butterflies shows that variation that leads to speciation doesn't necessarily need to come from new mutations, but can come from introgressive hybridization. And this may possibly accelerate the evolutionary process and therefore shows that hybridization may have a significant role in evolution. So the second question I looked at was, is speciation a continuum? Or is the species boundary distinct? And so allopatric species divergence is a great example of this. And allopatric species divergence can be seen here, where the species boundary becomes more distinct over time. So this is just showing the two species diverging and just questioning, at which point do you consider them a new species? And then hybridization, so a restoration of symmetry, reflects actually a continuum of stages. And so that just means that the genetic changes on the path to speciation, on the path to reproductive isolation, is continuous, and there isn't necessarily a distinct boundary. And so some evidence to support this was found in some research done by James Mallet, and he plotted, he showed a comparison of population divergence that reflected this continuum. And so here you see a graph, and it plots the number of hybrids against DNA divergence. And so you see that there actually is a correlation between hybrids and DNA divergence between two species. You see that as the number of hybrids decreases, there's an increase in DNA divergence. Or another way to put it is, as reproductive isolation increases, you see a divergence, an increase in divergence. And so this just shows that incomplete reproductive isolation that allows hybridization does have an impact on the genome. And so we see first that, in conclusion, adaptive integration shown in these Heliconius butterflies shows that hybridization does play a role in creating that genetic variation that leads to speciation. And two, we see that hybrid speciation reflects a continuum, and that there isn't necessarily a distinct species boundary when looking at hybrids, specifically in these Heliconius butterflies. And so in conclusion, Heliconius butterfly hybridization data shows that hybridization does play a significant role in evolution.